Body. What is it?
The Fleming of St Mary's Hospital, London, who discovered that it produces the drug known as
The
penicillin.
question was, which drug would go with which cell?
It's a collaboration that's unexpected because it's between humans and the tiniest organisms.
The howl of a dire wolf.
Dorset lamb may represent a major landmark.
I did not start off planning to design proteins.
Scientific breakthrough will probably be something that we hard to imagine.
Test at birth could map out an individual's entire future risk of disease.
Okay. So, tell me about your MD journey. What was it like? Obviously, you went to Harvard Medical School. Why did you decide to leave after 2 years?
Yeah, it's a great question. Well, it wasn't after 2 years. I'd say I probably left after 6 years. But yeah, I mean, the sort of reason is I think my whole life I had kind of been excited about getting into medicine, right? Like I thought it was one of the grand challenges of our time, right? Like we're starting to understand, what DNA is and cells and, how mutations can lead to disease. And it felt like it was just kind of an open opportunity with a lot of cool biology to try and solve it all. And I thought part of that meant like learning how to actually practice medicine, see patients, see the disease up front.
What did you want to specialize in? I don't think we ever really. Did you know what you wanted to specialize? You obviously told them when you got it.
Originally, I thought I would do internal medicine because it seemed like all the smartest people I knew would go into internal medicine because I had all the hardest puzzles. Like you grow up watching like House, right? And like there's House and like Grey's Anatomy,
doctor,
right? Who do you want to be? Do you want to be like Grey and Grey's Anatomy, or do you want to be like House, getting the hardest puzzles and like everyone like looks to you for like the hardest cases, right? And I was like, that's really cool.
I think Omar would probably be like, it's like Scrubs is probably the most accurate. Having seen him at the bench,
which is coming back, by the way, the whole cast. It was just announced. Yeah. So I think that was kind of the impression I had, right? And you don't really know what medicine and the practice of it is like until you're actually like in it, right? And so when I get to the MD PhD program, right, I do the first 2 years of medical school, which is great. I mean, it was actually really hard. It's funny. I did like mechanical engineering and bioengineering at MIT as an undergrad, and I thought that was way easier than the first 2 years of medical school because it was just like pure like here's literally textbooks you have to memorize, and like in 2 weeks we're going to test you on it. Whereas like at MIT, you literally had cheat sheets and everything. You never had to memorize anything. It was all about like, how can you from first principles reason through things, and you can have a cheat sheet, but you still have to figure out how to like solve a hard problem. And I found that way easier than just like memorizing a ton of stuff and having to like work your way through that knowledge. And so I actually, although it was a lot of knowledge and it was very helpful, I didn't actually enjoy the first years of medical school. I was finding myself wanting to be in lab all the time. And then when I finally was in lab, I was like, oh, this is like heaven. Like now I'm like tackling research problems, and we were working on CRISPR, which is a crazy story in itself because we were in Feng's lab at the beginning of CRISPR. And so I think we published like 30 papers together during that period of time. It was like literally the lab was like buzzing with like energy and ideas, and it was like back to back, like just competition, kind of like what you see with AI today, right? Except like there was like our lab and George Church's lab and Jennifer Doudna's lab and all like these great labs trying to like publish all these great papers. And so it was quite a journey. And at the end of our PhD, we still had so many ideas and things we wanted to do, and we were starting companies with Feng, and it just like did not make sense for me to like go back and like be in the hospital grinding at that point. So I kind of like went on a leave of absence, started a lab with Jonathan. MIT gave us space to like just explore cool ideas, which was fun. We started this company Sherlock with Feng around CRISPR diagnostics. And I kind of said to myself, okay, when the med school comes back knocking, they said that I would have 2 years to figure stuff out. I was like, when they come back knocking in 2 years, if I'm like loving what I'm doing and like we're doing great, I'm just like gonna leave and not go back. And turns out after 2 years, like we got a bunch of grants, like like multi-million-dollar grants from NIH. Like our lab was growing, we were publishing, and I was just enjoying pushing the frontier of science. And so yeah, that was kind of, I just like left and never went back.
Yeah. And you talked about wanting to be like a House MD type character when you first started. I think even that is being eroded as we speak, right? Like there is no, medicine as a practice is becoming intellectually outsourced to AI. There's no mystery that a human could solve, even a very, very smart human, that an AI wouldn't be able to solve. Do you think there's still a place for clinicians at all in society and in biotech specifically?
It's a good question. Yeah, I mean, it is like, Grok 4 just came out this week, right? And like it literally crushed the AIME like math examination 100%, like the hardest like problems you could think of, right? And like on like medical and humanities last exam, it does very well in those benchmarks. And you're seeing some of the stuff from like Microsoft, for example, where I think it was like 98% accuracy or something on like the hardest like medical cases. So, you're starting to see hints of like where AI actually can do very complex differential diagnoses. And so I do actually think it's interesting because like 10 years ago, I would not have guessed that would be the case. And now I'm thinking, well, yeah, definitely good thing did not stick to medicine. But is there a role for clinicians in today's society? I mean, I still think you need like a human touch, right? I was thinking about this the other day. Like, even if you had an AI in your pocket, right? Like, are you going to like let it automatically prescribe medications? And then when you do prescribe those medications, like what if the person gets a rash? Or what if the person wakes up the next day and is in the ER? Like, is the AI gonna like be able to do like longitudinal care for years for like very complex people that take many complex drugs? Like, we don't, like when you think about AI, right? Like it's garbage in, garbage out, right? Like you have to think about also the data you're putting in. And we don't have data on like complex medicine, right? Like if someone's taking 10 drugs, you have no idea what the interactions between those drugs are. You have no idea, like could there be liver toxicity? Could there be kidney toxicity? And so when I think about like, how do you like manage that? That seems very difficult, right? So I think for the foreseeable future, it's going to have to be sort of a combination of AI and doctors working hand in hand. I think you see that with open evidence, like taking off, right? Like doctors are fully embracing of AI. Like I think 30% of physicians in the country use open evidence, and that's like incredible. But I think the question you're getting at is like what happens in 10 to 20 years, right? Like when you maybe do start having that data and the AI simulations do get that good. And I mean, maybe that is one of the professions that goes to the wayside when you really can have a doctor analyzing you 24/7, your wearable data, your health data, your voice, how you walk throughout the house, right? We were thinking about this the other day, like, can you have ambient sensing in your house where it's like constantly just tracking you, right? Like you can tell your mental state by how you walk, your cognitive score by how you move, right? Like I think there's also like a lot of stuff that's going to change in the coming decade, and that I think is really exciting.
Yeah. I don't think it necessarily replaces doctors. I mean, if you think about like, it'll certainly augment doctors. I mean, the amount of doctors is really capped right now. If you look at healthcare spend, it's bloating, and a lot of that is like non-doctor roles doing things like, nurses and nurse practitioners, like practicing basically medicine on people. I think it will definitely, like even now, right, doctors use Google, they use UpToDate, now they use open evidence. So it's going to augment the profession. I don't know if it'll certainly replace it. Doctors may be able to serve more people, but there'll also be more people. But it will change the practice of what being a doctor is, right? Just like coding tools are changing what it means to be a software engineer. And it's kind of interesting. Not exactly a perfect metaphor, but there are more coders now because of these tools, right? There's not going to be more doctors because people won't be, Ronin doctors, but it will allow more people to function like doctors, not necessarily replacing doctors, but augmenting that. So it'll be complex. But what it will mean is that there will be more data and more predictions about things. I mean, a lot of, I think, medical decisions are made without a lot of personalized data and without a lot of these insights that a machine learning approach could have, right? Drug personalization. They give you, a doctor prescribes a statin. Are they going to pick like lovastatin or another statin, right? What is that based on? Well, it's probably based on some sort of heuristic they have in their head or something, or this works, or maybe, I don't know, I like that pharma rep or something. There are personalization approaches that you can use, knowledge graphs and a lot of these like virtual models of biology to improve that. And so I think that's where it's going to be much more personalized and much more effective, and much lower latency, which will be really nice.
I always like to like do a temperature check. Like there's a reminder, like AI is like not magic, right? There was a benchmark going around today where like it was a picture of a hand and it had 5 regular fingers and like a thumb, right? So 6 fingers. And OpenAI, Gemini, Grok, you ask it, how many fingers does this hand have? They all answer 5. Right, so like there are still like major gaps like in understanding, and like I don't know if I would want that thing taking care of me when I like end up in the ER in the middle of the night. Like I probably want like someone who actually knows what they're doing, right? And so, but what I do think is interesting is like something Jonathan said, which is like, what if you could have a doctor whose practice is like 100,000 people or like 500,000 people, right? Because like the AI is doing most of the work, and then just like the edge cases are what show up on like your desk every day, and that's what you handle. And I think that's really interesting, right? Like you could get like tech scale, margins and scale, but for something that has never had that, like primary care or what's.
Yeah. The other thing is like medicine is practiced at different tiers in society, right? Like the PCPs that we go to are different than the PCPs that Jeff Bezos goes to, right? Concierge medicine is a huge area, and we're starting to see kind of this democratization of this where blood testing companies, which are essentially sending you to Quest or LabCorp and getting a bunch of vials of blood taken, can do these large panels. But there's this question of what other information can we start to use using machine learning, right? Your voice can tell you or be indicative of your blood glucose level, right? Maybe we should analyze this later and see how much I ate. Your face obviously carries a lot of information about your BMI, but also stress, sleep, cardiovascular health, right? So there's a lot of information, I think, that will start to actually be able to augment and so give people more of this concierge-like medicine approach. So there'll be more care and ideally be better care enabled by machine learning. And for that, we'll still need doctors, but they'll be able to interpret this information, this richer information, to find things earlier, to look at your wearable data and say, okay, you have some sort of potential mental health or neurodementia hypothesis, or you have an undiagnosed metabolic disorder. I think that all will become more and more possible with more data and better models.
Got it. And Jonathan, obviously, you were also interested in biological engineering, but you met at Feng Zhang's lab, both as first-year grad students, right? So talk me through, how did you decide that this is what you wanted to do? How did you decide that you wanted to join Feng's lab? And how did you guys meet? What was that first interaction like?
Yeah. So actually, we met in undergrad, and that's a whole different story. But I think in terms of doing kind of tools and CRISPR, and I think that it's this classic Sidney Brenner, things are driven by tools, right? What is it? Tool development, great discoveries, in that order, basically. Butchering the quote, but tools drive a lot of things, and I think that's applicable outside of biology or even the sciences, right? Going back to machine learning approaches and tools, and hard tech has driven a lot of downstream things. And so when I was deciding about what I wanted to do, when I got to college, I was very interested in biology. I thought it was a very neat space, but it's what do you choose, right? How do you pick what to study? And actually, it's funny, I remember this was the summer before I went to college, and I was listening to a pancreatic endocrinologist talk about why he likes the pancreas, right? And he was like, well, it's the only organ in the body that can dissolve the whole body. And and he also gave another example of optometrists or other eye doctors, they love the eye because it's the only place you can see an exposed blood vessel in the whole body. Those are completely arbitrary things. It's how do you choose what to study and what to do? And I think as you kind of go back on the tech stack, so to speak, tools drive those things, right? It's kind of a punt to say, okay, how do I optimize my impact? Well, I'm just gonna go towards the root of things instead of the leaves. But that was, tools, right? And when I started grad school, I wanted to do tools, right? I rotated in labs doing optical physiology, right? And basically optogenetics. Did some DNA origami and some microscopy approaches, structural validation or structural elucidation using kind of molecular tweezers and stuff. And and then CRISPR was obviously something where it was broadly applicable. You see something that takes a hold of a field and spreads so rapidly, you just know that that's an area where you'll have impact. And so that was the place to be. And also, it was a really dynamic environment. As far as Omar and I met, we met in undergrad, we pieced it together on a bunch of biological engineering classes. We actually did a bunch of, harebrained isn't the right phrase for it, but maybe it's one that works. Like we did a project together where we were cooking up something like Ed Boyden's lab on, can we do protein structure prediction, or not protein structure prediction, but can we sequence proteins, primary sequence, by like basically spinning them on a disk that denatured them, stretch them out, and then do basically atomic force microscopy or electron microscopy to like read out the individual residues. Crazy stuff like that. We had a senior project together. So we were kind of pretty aligned about like, okay, let's try some crazy moonshot stuff. And when you're an undergrad, I mean, at least back then, none of that stuff works. But then when you're in grad school and you can be like, oh, crazy stuff, you can start to cook stuff up. And so, one example is that in grad school, we always were like, how can we develop tools to do insertion into the genome? And I remember we drove up to New England Biosciences to like ask them all about like ligases. Can you ligate into the genome? And now it's, we've done multiple technologies in our lab about large insertions into the genome. So I guess that was a fun little like 8 to 9 year journey. So yeah, I think when you find someone you can brainstorm with and when you get aligned with on like the outcomes and that you can bounce ideas off of, it works well. And we figured that out, I guess 15-ish years ago, maybe like 14 if you want to be conservative. And it worked pretty well. So yeah, been doing that kind of ever since. But the thing was we chose Feng's lab kind of independently. So that also shows that we're aligned. And we didn't coordinate on that. So yeah, haven't looked back.
Interesting. Let's talk a bit more about tool development, right? I think you and Feng, David Liu, Ed Boyden, probably some of the only PIs that consistently successfully do tool development, which is, I think, a testament to the fact that it's like a difficult thing to do. It's a lot of manual labor. It's kind of a little bit blind at times, right? But before we go into each of these tools, what is the current genome engineering toolkit? Like what are the tools that we've developed so far? What's broken about them? What needs to be fixed?
It's a great question. Yeah, I mean, there's been multiple generations of, one, genome editing tools, and then at a higher resolution, CRISPR tools, right? Genome editing dates back to, I don't know, the 80s, 90s, when you have, restriction enzymes, discovered that can manipulate, DNA and cut them up and allow you to do molecular cloning. I would say like that's kind of like the first hints of being able to do genome editing, where you can actually like start manipulating DNA and like doing recombinant DNA and like, manipulate biology, right? And that leads to this whole space of different types of nucleases that you can actually use to manipulate genomes inside organisms, right? Like meganucleases and zinc finger nucleases and, talons that all came before CRISPR. And what was unique about CRISPR, up until then, everything required fairly heavy protein engineering to reprogram to new sites, right? Like all those proteins I mentioned would require months of tinkering with the protein to program whatever site you wanted. CRISPR comes on the scene, right? And it's like, okay, this novel, bacterial defense system has sort of moved the programmability away from the protein level to a piece of RNA called the guide RNA, which is just complementary to the site you want to cut. And that's revolutionary because it allows you now to just very programmably from like your computer say, I want to target this site and this gene in the genome. You print that RNA sequence out, with an RNA vendor, and you're ready to go in like a week. And you only have to test a few designs, and most of the time those designs work, right? So it's almost like coding, right? It's like you know what to do, you do it, and it like generally works, right? And that was great. That caused the whole, CRISPR craze, as we call it. But ultimately, the first generation of CRISPR is actually quite limited, right? All it can do is it's cut, right? It's like scissors in the genome. And, that's useful for destroying bad proteins, so like proteins that are mutated and then aggregate. You can think of diseases like sickle cell, or, there's like fibrosis, like ATTR, which is what this company Intellia is going after. But there's 7,000 genetic diseases, and most of those actually need very precise edits to correct the mutation that causes those diseases, not just a blunt scissor. And so, groups like David Liu's lab developed, second generation things like base editing and third generation editing called prime editing, which are fancy ways of using CRISPR to now atomically change the DNA to exactly what you want. You can change an A to a G, a C to a, A. You can, not even do single base edits, but you can replace small stretches. You can delete, you can insert. And then, our group and, David Liu's group at the same time even went further to allow you to insert entire copies of genes, like thousands of base pairs, this whole idea of programmable gene insertion. So now you can go away from just replacing mutations to actually replacing whole genes or, even loci, or maybe one day we can actually replace whole parts of chromosomes or entire chromosomes themselves, right? That's like the vision that I think genome engineers want to get to. The cool thing about being able to do gene replacement is, when you think about a genetic disease like cystic fibrosis, there's thousands of mutations. And so if you're going to do genome editing and make thousands of genome editing drugs, that's really difficult, right? You'd have to essentially be making a drug almost for every small, unique patient population. Whereas with gene insertion and gene replacement, you can actually have a single drug for the whole, patient population. So that's really powerful. But what I'll say is, despite having this whole array of technologies now, and there's even multiple, versions of base editors. I think base editors go up to version 8 now, version 9. Prime editing is similar. Gene insertion is becoming similar. It is still very difficult to get these systems working, right? Every disease is a unique cell type, a unique cell state. And when you put these components in those new cells, they're not as active. And so you have to engineer and iterate. And sometimes it just doesn't work that well, right? Like when you put a small molecule in the body, every cell gets that small molecule, right, for a drug. But with genome editing, not every cell is going to get your genome editing molecule. Not every cell, even if it gets it, will get edited. And so you have much lower efficiency when you develop these drugs. And so there's a lot of progress that needs to be made on just making genome editing tools simpler and more efficient. And then also like working on the delivery problem, like how do you actually get these complex machines into like the exact cell type you want, which is not a solved problem. So that's I would say that's the scientific state of the genome editing space. The commercial side and does it even make sense as a modality is a whole nother thing. Maybe Jonathan can cover that if he wants. But
yeah, I know. I mean, like you think that it's difficult right now because it for rare disease it can take a lot of like when you're thinking about which indications you want to use a technology for, you have to think about what's the development path, right, and how long will it take, and what can we do? And I mean, there's a lot of caveats here where, one, what are the diseases that we can target? So 7,000 rare diseases, which of those can we deliver to? Which of those have, mutations that cover large segments of patients? Which of those have windows of therapeutic interest? And so it actually filters it down. You see a lot of companies kind of going after the same sets of diseases, right? There's certainly not 7,000 different, diseases that these companies are targeting. So we need better ways to deliver. We need better ways to, make therapies quickly. I mean, N of 1 initiatives, which until very recently were really based off of things like antisense oligos, have started to take place in the CRISPR world, where, I guess baby KJ was a great example of very quickly developing a therapy for essentially N of 1 case. And now with the recent Chan-Zuckerberg gift to the IGI, with the recent Chan-Zuckerberg gift to the IGI, we'll see more of that. But yeah, I mean, it's hard when you're targeting rare disease, especially in an age of biopharma where everyone wants to like treat cancer or obesity. So I think one big thing actually, like for the field, I mean, what are the ultimate challenges for genome editing? And it's 3 things. One, I mean, make the tools themselves better in the absence kind of delivery, which is hard to say. Make delivery better, which means like, can we use AAVs? Can we do protein-free approaches? In the lab, we have a protein-free nucleic acid editing technology that we like a lot. But can we deliver them better? And then I think the third thing is, can we start to apply them to complex disease? I think that Verve is a great example of this, where taking a base editing technology and applying it to a complex disease. There you have a target that has other approaches towards it. So there's questions about, is this the best way to target it? But for targets that are important in complex disease, like neurodegenerative, cardiovascular, autoimmune, cancer, obviously, from probably the immunology side, can we use genome editing approaches or nucleic acid editing approaches to treat these? And I think that is a wide open space, which will come with much more understanding of biology. both from kind of genetics, genomics, and other, probably machine learning approaches. The three very different problems, and a lot of, focus from those labs you talked about in the tool space, still really is kind of on that first thing. How do we make the editor better? But how do we deliver better, and how do we actually find new targets and find new ways to approach targets? I think is a huge problem. It'll be solved, but I think you're underserved areas, and we need to speed those up as well.
What is the dream publication here? Like the final end state enzyme or RNA based enzyme that achieves your goals, and that you could sort of sit back and say, I'm satisfied, we've solved genome engineering.
I don't think there's going to be one enzyme that solves it. I mean, I think you can't have one enzyme that like just wins, right? I think we have to look at it disease by disease basis. And so like the final thing is that, can we apply something to cure, like, for example, one of these entities, like, can we cure all PKU patients, or, can we do sickle cell without having to do like actual conditioning and take cells out of the body, like in vivo sickle cell, or more complex things, right? Can we actually have gene therapies or nucleic acid editing approaches that treat cardiovascular disease, that treat neurodegenerative disease and dementia? I mean, those are like really, as you go up the hierarchy, will be huge things. And I think there's a lot of these things. And eventually, our lab likes to think about longevity a lot, whatever, that loaded term means. Can these actually, treat multiple diseases through one therapy such that you get a overall extension in kind of, health span or, quality of life adjusted years. And then I think those will be very cool. So I would love those. Publications, a publication is a publication. I think, but thinking about what the impact on patients or, just people is really, there's a lot of those big things left. Too many to count, probably.
Got it. Yeah. Interesting. You have something to add?
Yeah, I think also, like, it may not even just be an enzyme, right? Like, I think enzymes are honestly not the best drug modality. Like, if you could figure out a way to do small molecule genome editing, I think that'd be way more effective, right? Like, imagine a small molecule that can be designed maybe with AI, right, to bind the exact DNA sequence you want, and then recruits the enzymes in the cell already to make cuts or make edits. And I think that's like much more of a scalable and tractable approach to, because once you're in small molecule land, it's much easier to deliver to any cell. It's much simpler, to make and manufacture, and it's yeah. Small molecules are great drugs, right? So, yeah.
Classic.
Definitely more scalable, but let's take the alternative approach of, these RNA-guided endonucleases getting so good, so small, they can fit in AAVs with all of the cargo that you need in one shot to all cells, and you can insert whatever you want.
Yeah, but like AAV doesn't make sense as a modality for genome editing because you overexpress a CRISPR nuclease for long term, and it's not great. And so I think any company doing AAV delivered nucleases is honestly doesn't make sense as a drug modality. And then AAV in itself is struggling right now. AAV just killed another person, I think like last week. Was it the Sangamo? Not Sangamo. Sarepta, I think. I forget which. I think it may have been Sarepta. Do you know what I'm talking about? Like, yeah.
Wasn't it sort of just too high of a dose?
Yeah, but that's the issue with AAVs. Is like they're not also that efficient at delivering materials. So you end up having to dose them very high, and then that's what happens, right? So like AAV still has a lot of things to work through, and also it can't deliver to a lot of cells in the body. Those all might be solved problems, but this is like the struggle, like just in general, gene and cell therapy is going through right now, right? Like, look at like weight loss drugs, right? Like you're not killing people there, and you're like completely transforming like a whole like part of society, right? It's like very different.
Something that would give me nausea, I guess, in terms of AEs. And the other thing is like, like terms of cargo capacity, right? Like thinking about, well, how will we ever like deliver like 10 kilobases of sequence? It's not going to be through an AAV. There's going to be different approaches that are necessary for different things. But yeah, in terms of like the protein side, obviously getting the efficiency up and up. So you need less and less, and that can help solve some of the delivery issues. Being able to do more complex edits may be good. But, it's one part of a puzzle or one part of a solution, I guess, where you have many different aspects of, how is the delivery being done? How are we delivering if we need a donor template? That piece. What's immunogenicity? What is just like the off-target effects obviously are a big concern. So I don't know if there'll be one editor that solves all of that. That being said, I think that, going away from like endonucleases and, some of the retrotransposon-based systems, I think, have some promise because those you could deliver potentially with an LNP as an mRNA system, and that could go and insert, maybe a little more sequence than an AAV-based approach. That's, something that could be promising. But I think there's probably not going to be a one-size-fits-all approach. And I would be shocked if there was one dominant technology that like led to cures, right? It's not like base editing or prime editing or traditional nucleases. There's not one winner, right? All of these different technologies are being used to treat different diseases or being developed for different therapeutics. So I think it's going to be kind of a much more cosmopolitan landscape of technologies than there's one dominant technology that, is used in spite of all the others.
Interesting. Let's take the 20-year from now future. Bridge recombinases or just like some other technology, maybe STITCHR, has led to the emergence of the programmable gene insertion therapies actually becoming approved by the FDA, right, for all sorts of things. LNPs get so good, you can deliver them everywhere, to every cell type, with lots of control. What do you do? Like, why should ordinary people, be excited about programmable gene insertion and delivery?
So ordinary people, I guess there's two ways to parse that, right? Like you could be excited like, oh man, this is going to be great. We could probably cure a lot of rare diseases with that approach, of course, maybe potentially all of them. But it could also affect common diseases like installation of protected variants or other, engineered enzymes. Like, could you produce antibodies in your body that then you could actually, or other enzymes in your body, insert those into safe harbor sites or other good sites in the genome, and those could, deplete proteins in your body so you wouldn't have to take shots, right? Making biologics in your body has been a dream for a long time, and it was Moderna's early pitch, and perhaps that can be something that is realized by that. I think that there's a lot of potential for medical enhancements due to that type of editing. But a lot of that depends on, it's not just solved by these tools. It's solved by, can we make better antibodies, or protein therapies that do these things? Do we know the biology? If those things are met, then yes, they kind of all come together in a really cool way. And then I think, hopefully we can use this to target those really complex diseases, especially once we understand the biology.
Kind of drawn out of that.
No, I think the only way it connects is through human enhancement, right? Like most people don't have a genetic disease. So yeah, as Jonathan mentioned, there's a few things that you could do that's interesting. I think other ones, right? Like there are people who sleep naturally for like 3-4 hours a night, right? And like are totally healthy, don't develop dementia or Alzheimer's, right? It's just like, pure, like they get an extra 4 hours to their day, no problem, right? And that's been tracked to like a bunch of different genetic variants, for example, right? So you can imagine like, could you install genetic variants that make you sleep for 3-4 hours a night and still feel like completely refreshed, right? There are like, genetic variants for like, not eating as much or like, better muscle, better bone. Like, could you think about stuff like that, right? I think you're starting to see a little bit of this. Like I would argue Verve, right? Verve Therapeutics is borderline human enhancement, right? Because it's like installing variants that lowers people's cholesterol, right? And they want to take that to like the common population, right? And like that's kind of interesting. Like you could just have a one and done drug to like just have low cholesterol, low LDL for life, and that will probably make you live a lot longer, right? So now you're like living longer from a single dose. That's kind of interesting too, right? And so are there more examples of things like that, right?
I think also there's going to be cultural things in terms of like, when you talk about like genome, like potentially permanent things versus like, would people prefer to have a therapy that, could be reversed or not, right? At the edge where it's like, there's no technical challenges, maybe there's still some sort of like considerations where like, oh, do I want to do this to myself permanently? Maybe not. People do a lot of stuff to themselves permanently right now even. But I think,
this is
wide adoption.
I think
is a big
that's
question.
one of like the biggest knocks on genome editing for human enhancement, right? Like with AI, there's like a new advancement like every month, right? And like this month I'm using Gemini, maybe next month I'll use Grok 4 or like Claude, and it's like I think biology will accelerate to that degree too. And it's like, are you like you want to like permanently install something that you're stuck with, right? When you could like not do that and have a ways to upgrade, right? So that's one of the things I like can't grapple or like wrap my head around exactly with genome editing. Like I don't know why you would want something permanent when you would have the chance to upgrade with other modalities.
The adoption curve will be, and it won't be everyone, but yeah.
Another example is I was on Ozempic for 3 years, right? And it was great, lost a lot of weight, but then I switched to Zepbound, which is the Eli Lilly drug, and I'm losing even more weight. It's like, and soon there'll be the next generation Eli Lilly drug, which is going to have like the muscle activin antagonist, I think, or agonist, yeah, that like basically preserves muscle and basically helps you build muscle, which is kind of cool, right? Like imagine taking a drug where you just like don't have to weight lift, but you just bulk up, take that creatine every morning, yeah. So
awesome. And you're both interested in aging, right? This is something that I'm getting at. Aging, lifespan extension. How did you discover that you each shared this passion? And for our audience's sake, why should we care about aging? Why is this the right modality or sort of framework to view complex diseases?
They're aging, because they are aging.
I think, maybe in a decade you'll understand exactly why should you not care about aging.
Wow, that's very patronizing.
No, I'm just kidding. But like seriously, I'm like mid-30s now, and like I get home, like my knees ache, and like I need like a massage every week because my neck is sore.
Need a massage. That's an exaggeration.
I think no, I do. It's I feel like you hit 30, like literally the day you hit 30, and it's like you start putting on weight, and like your metabolism slows down. So like now I'm starting to get gray hairs. He has way more gray hairs than me. So like I think you start to see your body change quite a lot, right? Like in your 30s and 40s, and a loss of energy. And I think there's a lot of biology behind that. It doesn't have to be that way, right? Like I think we all want to live healthier, longer lives, more time with your family, your loved ones, more time to enjoy life and be productive. And I think that's why everyone should care about it. I mean, if you look at the next generation, like Gen Z, like they're spending way more on health than any generation before them, which I think is really interesting. And they actually drink substantially less. Like there's this whole trend where like people are like basically not drinking alcohol. And I think that's really interesting. I don't know where that health pilledness comes from. If you could figure it out and bottle it up, that would be great. I think partially it's social media. Like you have influencers and everyone peddling good looks and healthy lives, and I think that spreads and becomes contagious. But I think that's going to be an increasing trend where like just more and more people are going to prioritize health. I think with AI, like there was some report I was looking at where it's like during recessions people spend more on like health and wellness, which I think is very interesting. And so I think with AI, where like there's gonna be more job displacement and probably more time on people's hands. Like let's fast forward. Like let's say you have like abundance, right? You don't have to work that much anymore. Well, then you're definitely probably gonna be spending a lot more on like your health and like your wellness and like living a better life, right? And so I think that's where aging, science comes in. I think that's where longevity interventions come in. Like we're just starting to understand how to reprogram cells, how to rewind the clock on cells. And yeah, I mean, I think that's exciting. I think the other angle is like there's some diseases of aging that are just so devastating, right? Like dementia and like Alzheimer's, right? Like just the losing yourself of who you are and like your memory. And it's like, that's really crushing, right? And like so also solving some of those things I think is really important. But that's my take.
Yeah, I mean, I think that overall there's a lot of reasons to think about it. One of the earliest things I remember when I wasn't like having aching knees. I was actually pretty young when I got interested in aging, and I think back then it was like sirtuins were a very interesting target. So I did a lot of sirtuin research. I mean, there's a common trope of like if you take top diseases where they found the cure for cancer today, well people are going to live 1 year later and then have like mortality due to CVD, right, or cardiovascular disease. How do you actually approach these things? Well, you cure aging, whatever that means. I think it's but like from a benefit to society standpoint, in terms of like if you can target, in one framing, the root cause of diseases, then you could make more quality years of life for people versus targeting a specific instance of one of these diseases. I think actually another interesting thing is that aging is still pretty hard to pin down as like what it is. And I think that's pretty unique. Well, relatively special among different biological concepts. And so that provides a nice challenge to say, well, how do we understand this complex phenomena when we can't really even necessarily agree to what it is? I think it's obviously hard, but that is a nice problem statement versus, there are some things where obviously in cancer, we have a fairly good mechanistic understanding of like what cancer is as a description, right? Maybe not at the basic, fundamentally understanding the mechanism of cancer, but like, what is it? Aging is more complex and longevity is a little more complex. So I think that's a neat place for these new tools. And then I think in terms of how we can like actually cure diseases, if you think about from benefit, like people like right now, like this pipeline in a pill concept, and like Ozempic's a great example of that, where, okay, diabetes, obviously weight loss, potentially some neurodegenerative effects, gambling, right? Like, it affects people and makes their life like much more improved. Those are the things that have large impacts on society. So maybe it's not extending people's to live to 200 years old, or maybe it's not even extending their lifespan at all, but even like a health span extension where I have a better, life. I have more quality time with the people I love. Those are all important things, I think. So it's, a challenging area, one that I think has made good progress and been exciting over the past couple of years. But obviously, one with huge impact. Also large ethical considerations, which I think, aren't something to brush away, but it's interesting to have that dialogue as well. So yeah, it's a cool area to apply new technologies to, whether it's new molecular technologies, whether it's new, computational technologies. I think it's one of the bigger unsolved problems in biology.
Like I forget which like goated founder said this. It may have been Steve Jobs, but like, the goal is to like build products that delight people, right? And I feel like you don't, historically you don't get that opportunity in biology, right? Like you focus on like very specific diseases that affect very like specific subpopulations of the world, but with aging it like affects literally every single person, right? And I think for me, the first time I felt something in medicine or biology like actually delighting me has been like Ozempic. Like it's like pretty crazy, right? Like you inject yourself once a week like in your thigh, and then like the fat just melts off your body, right? I lost like 80 pounds, and you see it like literally every week, like just like your face, your body, your scale, the clothes that you haven't touched in like 10 years in your closet, you're like dusting off the cobwebs and you're like, oh, hello again. Literally rediscovered shirts I had, I forgot I had. So
Omar's like fashion sense is like a time capsule, basically. Justifying that.
So like I think that's like very cool. Like, and so like, what else can we do in biology that delights people? So like, I'm not even as interested in like the medicines per se, right? Like we talked about like, can we make stuff that like helps, like lets you build muscle, right? Or like literally simulates exercise on your cells. Like, imagine you could like exercise 30 minutes every day without exercising, right? Just like another pill. Or like I said, like a pill for like Ozempic for sleep, right? Like where you could take something and sleep a lot less and be just as, effective. Or imagine we can make like a nicotine that actually is not addictive or bad for you, right? Like, I don't know if you've seen the trend of Zins, but like Zins are like incredibly, one, popular and two, effective. Like they really give people better productivity and focus for work. Like imagine you can make like actual versions of that like are real, right? Like this whole idea of nootropics, like can you control, different aspects of the brain, like mood, for example. Like we can't control emotions. Like imagine you actually could control your emotions. That would be like also transformative. What?
I mean, people can control their emotions to an extent, but yes. No, I mean, I agree with your point.
Yeah.
Directionally.
Jonathan's part of the population that thinks depression is not real, right? It's like... No, like there is an aspect
I agree with you on that.
There's an aspect like it's hard to control.
There's definitely an emotional control. No, yes. I think like, mental health, like, sleep, wellness, mood, happiness, like there's a lot of these aspects that, biology can help, right? I mean, I was gonna say the first thing that you've taken that makes you feel happy. I mean, hopefully you've taken a painkiller before when you've been unhappy, like, but also like non-addictive or non-opioid painkillers, like effective medicines, like those. I mean, there's so many things in your life that, you know, could be improved, even for people like who aren't suffering from, you know, a chronic disease. And then of course, people with chronic disease or you know, pretty substantial elements, it applies to them too. So yeah, build things that are impactful, and agents are one kind of shortcut to that.
Like as you know, like for example, like we have a project in the lab where we've made every secreted protein and peptide, right? And like we screen them for different things, and like we're starting to see very interesting hits for things like muscle growth or, you know, even skin and, you know, other types of these things. And there's so much undiscovered biology just like waiting to be found. And so I think if you can map these types of things out and find new ways to modulate these phenotypes, it's really tractable. Like it sounds, I think, very abstract and high level when we talk about it, but there's actually real ways to break it down to molecular pathways and things you could actually build as a drug or product one day, which I think is exciting. I think the other angle, you know, is using AI. So like we've actually also been collaborating with Google, for example, to use like advanced AI systems to also nominate, you know, pathways and interventions that can modulate these things as well. And I think that's really exciting because you can go very quickly when the predictions are such high quality towards an answer for these things. And I think that's another area that will be interesting to watch in biology.
Yeah, I mean, it's funny when you talk about, I think even Google said today that they're going to try to cure every disease. You know, you look at these large language model labs, like, you know, OpenAI, Anthropic, DeepMind, and they've all said that they want to use this to cure diseases, right? I mean, obviously DeepMind is, you know, doing that in a lot of different ways, not necessarily with LLMs, but like, you know, isomorphic lab is big. I mean, Dario, Anthropic used to be a biologist, right? I mean, I guess technically he's still a biologist, but, you know, so his PhD is on. You know,
And Demis.
Yeah. Yeah, well, yeah, and I mean, Demis is a Nobel Prize in chemistry. You know, all these things can be applied to, I think one of the big applications, aside from, I guess we never have to work again and we can, you know, generate our own movies, is that, you know, we'll cure disease using AI, right? It's like very, like, cool as a biologist to see that as trumpeted as this big application. It's funny, actually, though, also that, you know, there aren't that many applications in that, right? It's like, you know, someone comes out with a new AI model, right? They're not like, oh, we use this to cure all diseases, right? It's not a big focus. I know that there was work on the Grok presentation on applications to biology. It was like, what do those mean, right? How do we understand it? So I think, you know, hopefully, and, you know, this is a big focus of us, how do we actually take these, like, you know, new foundation models and apply them to biology? And how do we train models, foundation models, to do better biology? It's, I think, going to be a big flywheel, big unlock, right? And that's, you know, we talk about tools, that's probably going to be this generation's most impactful tool. Hopefully it bears fruit, just like CRISPR did.
Mm-hmm. What I'm hearing is a lot of human capabilities enhancement. We've talked about Ozempic for sleep. We've talked about small molecules and peptides being sort of more scalable, well-trodden modalities to bring these benefits to patients at large. But we've also briefly touched on the secreted peptide project, which I think is a really cool project because there's currently no systematic way of screening all the potential GLP-1 agonists out there, and you guys have come up with something. So tell us a bit more about that.
Sure. I mean, we trade off on this one. I mean, I think that, you know, screening technologies are unbiased screening technologies or minimally biased screening technologies are a great way to understand biology, right? A genomic screen is I have an outcome, a biological outcome I want, like cell division or, you know, killing a cancer cell or, you know, translocation of this protein from this cellular compartment to that cellular compartment. And I can throw genomics at it, or genetics at it, sorry. I can throw genetics at it to figure out what's involved, right? This predates all of these pooled screening technologies. This is how biology was done, like, you know, for decades, where let's create genetic variation through mutagenesis or breeding or whatever and figure out what that is. Now, of course, now with, you know, pooled screening technology like CRISPR, we just knock at every gene in the genome, figure out what genes led to the phenotype that we want, you know, to the T cell, you know, getting penetrance into the tumor and killing, you know, it. and we're golden, right? I mean, it's obviously a gross oversimplification, but these are powerful and biased technologies to like do discovery in a less hypothesis-driven way. But it's also very interesting that, like, you know, we have all these cool aspects of signaling biology. There are, you know, amazing signaling drugs like GLP-1 agonists coming out, and you actually can't say, okay, well, what's the effect of every, you know, protein or peptide that's secreted by cells on a, you know, on a phenotype, on muscle growth, on, you know, T cell exhaustion, on immune states, right? And so, and that's hard because you can't like knock those genes out because it's something that comes from outside the cell, right? When you do a lot of these assays, they're in a pool, and if you're secreting peptides or knocking out peptides, you have all this crosstalk. So we came up with a solution to that, which is, you know, there are evidence of this in the literature before. People, you know, can tether a peptide or a protein to the cell surface. And so this has been tried in different varieties before. You know, in some ways we just scaled it up and kind of made it a more easy to deliver library. But we can deliver thousands of secreted factors, and this can be things that are in the genome. It can be predicted factors. It can be machine learning developed factors. And we can tether them to the cells, so they only affect those cells in a self-signaling and autocrine fashion. And then we can say, does this make your muscle grow bigger? Does it, you know, make your hair cells proliferate more in terms of, you know, aging hair, right? Your baldness. Does it reduce inflammation in a like immunostimulatory model of like, you know, immune cell or myeloid cell inflammation? It's a broadly applicable platform, and I think it's very cool because we can start to see, well, these factors have these impacts. So it's another tool in the toolbox, so to speak, but it opens up this kind of area of like secreted biology that we hope can be useful. Other efforts, I think, are underway where it's like, okay, mapping effects of different light ligands on GPCRs, for example. So the FRO Eve had a really great bolus of data that they put out where they're looking at basically huge amounts of small molecules versus GPCRs. You can imagine huge amounts of protein and secreted factors versus GPCRs. And this starts to create a much more like, I think, complex interaction network that we can then use to discover. And it will be useful to train models too. So yeah, we're very excited about that. I should have given you, build upon that. I should have carved in a card.
I think that was a great summary. Yeah.
Well, I think the challenge here is that usually these peptides, endocrine signaling, right? It's hard to capture the effects of these peptides on an organism without, you know, actually studying a whole organism over a long period of time. So this is, I guess, and you can correct me, but somewhat akin to a peptide version of Perturb-seq, right? You are in a very scalable, high throughput way being able to assess different peptides' effect on the readout of each particular cell or all of these cells by literally tethering the peptide that you've designed, whether it's like an engineered peptide or a natural peptide, to the cell surface, and then seeing what effect this has on the RNA readout, right? The single cell RNA-seq readout.
Or whatever. I mean, it could be RNA, it could be, you know, does the cell divide, like, you know, growth, right? That's the simplest thing, growth or viability. We can stain for a marker and then do like something like flow cytometry, so we can look for like biology on that end. I mean, RNA-seq is obviously like probably one of the most powerful tools you'd have to read out of, but something's a little too powerful, I would say. And you know, I think you pick the right kind of readout for what you want. But Perturb-seq, of course, like being able to do that with a peptide like library, you know, would give you a lot of picture.
Yeah. I think there's all sorts. You can get as creative as you want. Like we've come up with a molecular frailty score that measures aging across a bunch of different, you know, molecular like assays and axes of the cell, and we have a screen where we're just like reading out all those assays, and then, you know, seeing what peptides can hit like multiple orthogonal pathways of aging, which I think is really interesting, right? Like everyone gets to like this question of like, is there a master peptide out there that can just like broadly rejuvenate a cell? And like, that's one of the questions we want to answer. It's akin to like, you know, think of like Yamanaka factors, right? Like you express these 4 transcription factors and you like literally reverse time on cells back, you know, to like a stem cell state. And our question is like, can you find things like peptides that can do something similar? And the reason for that is it is very difficult to turn Yamanaka factors into a drug. You're not going to deliver 4 transcription factors, including one that's oncogenic, and has been shown to cause tumors to people. And then it's very hard to deliver them anyways, because you have to get them inside cells and then into the nucleus. And so if you can find, better things, whether it's small molecules or peptides or whatever, that could be one of the holy grails of true rejuvenation. So, yeah.
Yeah, it's likely that there remain many GLP-1 level drugs that remain undiscovered because there's no high throughput way to assess them, right? This would be great for the lab. Obviously, lots of funding if a drug comes out of this. But let's kind of go back to AI a little bit, right? So another paper that came out of your lab was EvolvePro, sort of directed evolution in silico. Tell us a bit about that, and like, what's the use case? Like, what was unique about this? And yeah, like, how can biologists use this tool?
Yeah. I mean, I think I would say, like, it's funny, like, now half our lab, is doing variations of AI in biology, right? Our goal has always been how to make biology programmable, and historically that was through molecular tools like peptides and CRISPR, and now it's turning out AI and machine learning tools are becoming powerful enough, partially because of exponential growth of different data sources in biology, but also partially because the models have gotten good. And so we have projects across every scale of biology, from modeling DNA, RNA, proteins to virtual cells, virtual tissues, and even virtual people. And EvolvePro is kind of in this bucket of how do we model nucleic acid essentially, and the problem and proteins. And the problem there is our whole career has been built around protein engineering, right? Like, you find proteins in nature like CRISPR, you have to like industrialize them and make them much more useful. It's pretty hard. A lot of things don't work. You're constantly failing. It's like 99% of experiments fail, and then one day you get lucky and you can get a paper. Our goal here was like, could AI accelerate that process? And I think we were hopeful for a long time that someone would solve it. Like, there was all these protein language models and de novo protein design tools like diffusion models, but it turned out all the de novo stuff didn't work that well. It was you'd screen hundreds or thousands of designed molecules, and maybe a couple of them might work, and they generally worked worse than wild type proteins in nature, let alone the state of the art engineered proteins. And so when we saw that, and we saw like paper after paper that was just like the same thing, we were like, okay, like, let's actually step in here and actually try to build something that we would find useful as protein engineers, or could actually bring our skill set of protein engineering to the masses, right? Because like most people have no idea how to engineer a protein. So our goal there was basically like, could we build machine learning models that could generally try to understand the mutational landscape of a protein, not from evolutionary fitness, which is how protein language models are trained, which is the concept of like what makes a protein like able to fold and be observed in the evolutionary space, but actually like the sequence landscape that promotes protein activity, which is usually hidden, right? Like, evolution doesn't actually evolve for activity; it just evolves for like, is this good enough? And usually that's like fine for a cell, but not for like an industrial commercial application. So what we did is we actually started with protein language models that learn evolutionary fitness really well. And we trained models on top that could take small amounts of experimental data and quickly sort of recolor the landscape, but for activity instead of fitness. So the idea is, like, if you have a CRISPR protein you want to engineer, we could use a protein language model to do zero-shot prediction, for example, of like a few mutants that we think would improve activity. You go test them, but that new data you have now can feed back in and immediately update essentially the weights or the landscape to be more in tune with activity rather than evolutionary observed space. And then you predict the next set. And we do multiple rounds of that. What was non-intuitive is we saw very rapid increases in protein activity from only testing 10 to 20 predicted designs per round. And that's huge because that's such low end protein evolution allows first of all, anyone to do this, right? It's very hard to set up experiments where you have to screen thousands or hundreds of thousands of things as typical direct evolution experiments allow. So being able just to like have, okay, I'm going to clone 10 sequences and test them. Okay, that checks the box of anyone can do it. Two, it brings down the cost quite a bit. It's really expensive to screen hundreds, thousands, tens of thousands of things. And so bringing down the cost, again, checks the box that anyone can do it. And then the third thing is it actually lets you do some interesting things, right? If it's so low end, it means you can engineer across many different things. So typically when you do directed evolution, you have to pick one assay and make a whole screen around that assay. With this, because you're only testing 20 things per round, you can actually have many different assays. So if it's an antibody, you can have target binding. You can have the solubility of the antibody. You can have the expression of the antibody. You can have the half-life of the antibody. You can, if it's a bispecific, you could be engineering 2 different target binding, right? So that's really powerful. And you could even think about doing even more complex stuff like in vivo data. Like imagine you engineer an antibody, but all the things are actually in vivo experiments, because it's really easy to throw 20 antibody sequences into mice and collect that data. So I think that's like the power of this concept of doing AI directed evolution, is it really brings down the design space and makes it easier for people to do protein engineering. Yeah.
Got it. Yeah, and I think this is very distinct from existing tools, especially ones that rely solely on biological computation or just like a pure wet lab, such as PACE, right? Very few people can set up a PACE experiment in their lab. It's very complicated. It's basically only David Liu and Kevin Nesvelt in the whole world.
Yeah, exactly.
I know you guys have probably tried, and it's also been difficult.
No, dude, it's so hard. We did not even try. Right? It's a daunting task in itself.
There's lagoons and stuff. Yeah.
Yeah.
But yeah, I mean, I think the goal is like you want to make tools that are as easy to use as possible. And what is easier than a tool that you don't need to change what you're doing at all, right? If I do assays, I just do those assays and feed that data in, and it tells me new things, right? I don't have to do a selection or something. I don't have to order a big library of DNA. I just clone what I would clone before. And so that's really, I think, like the space of like anyone can pop it in, which is why I like people like these de novo binder approaches. As they start to get better and better and they can actually use them, okay, I'll just order this and use it. They don't have to do phage display or something else. Those are the types of technologies that I think will make it to the bench much quicker than bespoke technologies or things that use like specific requirements.
Yeah. I think that's a really important point, because like the more AI can simplify biological workflows, the more you can standardize those workflows, right? And that's really important because, like, for example, this EvolvePro, we're trying to develop some, for example, autoimmune antibody drugs now, and we don't actually need a lab space to do that, right? So we can actually work with CROs where you literally just have the AI design the molecules. The CROs run them through all the assays they already have set up, and you just feed that data back in. You do that active learning loop over time, and I think this concept of basically having virtual biotech companies could become more and more common, which I think is really exciting, right? Like imagine, right, you don't have to tinker anymore, right? Like if the science becomes less complex, you don't need to be that lone person in the lab just tinkering around until something, you have that eureka moment. And that's cool because like biotech's really hard. It's really expensive, right? Especially when you need lab space and equipment, and the timelines are long, which makes it even more expensive. And so if you can virtualize the lab, that actually helps to bring down that burn, makes companies look a little bit more like tech companies, right? Where you have a bit more of that margin and scale and runway. And I think that could be very good for the industry because like you can then have more people developing drugs, right? You can have more drugs coming to market. That's better for patients. It also increases the cycle time for biology. This is one of the big issues, in my opinion, with biology. Like in tech, the world changes every week, right? You just have such a fast iteration time, right? Things can change more quickly, and you have that compounding effect, which with biology, the iteration times are so slow, and that just makes everything slower, right? You lose out on that compounding effect, and so that's one of the things I'm excited about AI in biology, hopefully changing. But yeah,
yeah, we kind of talked about AI augmenting humans, making everyone some version of a doctor, making humans or everyone some version of a protein engineer, right? And eventually leading to maybe like a one-person unicorn in biotech, which is something that's like currently unthinkable, very, very difficult. Probably much closer to being able to get there in software right now. Two more things that I wanted to touch on this EvolvePro system is like this can also be done in mammalian cells. You can test activity within human cells if you'd like. B, right? You're sort of feeding this data in a loop. It's getting better at making these predictions over time. These actual, like the base models will become good at one shot predicting how to improve this enzyme or whatever it is. And then C, right, we're improving activity along a broader informational landscape, right? It's not just, can you improve activity? It's like, can you improve solubility? Can you descend down gradient descent, but not just in one dimension, right? Across multiple dimensions. And that's something that's like very fascinating about EvolvePro, right? Just like switch back at me anything that sort of wasn't accurate or.
I mean, I think you can do stuff in mammalian cells. You can just also do things in animals, right? Like try to do direct evolution of like, actual phenotype like tumor ablation, right, with a antibody, right, through an animal. So you could actually say, I'm going to put 20 mice or times 3, let's say 60 mice, and I'm going to inject my compound and see what works best, and I can iterate back and say, okay, these are the actual things that led to tumor loss or immune cell reactivation and things that you wouldn't be able to do in a screen format at all. Because it's not a simple assay. It's not in a, so that I think is something that's pretty cool that we're excited about, like just very, very complex assays that you can iterate on, or when complex, I mean from a labor intensive that would never be amenable to screening. I think we hope that more and more people use it for that as well. Yeah.
We've talked about data and AI. I think like a good intersection of that is virtual cells, right? So you guys have both talked about creating virtual cells to model aging and eventually using these to understand exactly what aging is and how we can sort of address it. For some listeners new to the idea, what is a virtual cell?
What isn't a virtual cell? It's certainly a very hyped term right now. I mean, I think what we like to think about is cells, and there's a lot of things that go into understanding what a cell is. But one useful heuristic for cells right now is expression, right? Where we start to think with like these large initiatives of single cell RNA sequencing, we can start to find this diversity of different cell types and states and potentially start to separate them out coarsely into pathogenic and non-pathogenic states. And now I would say by a far shot, not every pathology is amenable to this, but I would say maybe there are certain ones that are. How do we figure out how to go from pathological to non-pathological, right? What are the perturbations that we can use to get cells from bad to good in a very reductive sense? And how do we build models to do that? Well, can we build models that can predict the outcome of perturbations on RNA sequencing identities of cells and use those to screen counterfactually different types of things in silico where you would never be able to do in real life, right? If I get an Alzheimer's patient brain and I'm like, well, how could I get maybe this microglia in that brain to be less, inflammatory? Maybe that's good for the pathology. I'm not going to be able to do a screen in that sample, right? Like that person is not going to be able to give me additional brains. So can we actually do that in a simulated fashion, use that to actually nominate new things, especially in age patients, going from an age to a younger phenotype at the RNA-seq level, I think is very exciting. So we want to build models that can do that prediction. Very simply go from one RNA state to another given a perturbation. Or if I have a cell and I put a perturbation on it, a genetic perturbation, I think is the most tractable. Small molecules are very exciting. How do I figure out what will happen to that cell? And yeah, this is an area of kind of that has been put under an umbrella of virtual cells, which is, I think there's a variety of different ways to simulate those. But I think it would be a big unlock and understanding. Another, especially where if we can do this, when AlphaFold 2 was developed by DeepMind, they folded every protein in UniProt. Didn't do a perfect job, but you don't have to. And then you can use things like FoldSeq or other computational approaches to do that. What if I could take, every perturbation, genetic perturbation, and put those on every cell in the body or every cell in the atlas? You choose your own cell atlas. Can I start to look at patterns and figure out what's going on and what's interesting across it? So can we start to build these resources for the community that we are doing these large scale, perturb seeks screens on every cell that's ever been documented. I think this will start to be very useful and uncover very interesting patterns in genetic regulation. It's early days for the field, certainly, and it's been a field that's been picking up, but has been around, in different respects for, quite some time. I think these new machine learning approaches and these new amounts of data, both of observational and perturbation data, will lead to many innovations, and how you already start to see how these lead to innovation. So we're very excited about the potential as a general resource, as a tool to understand these complex diseases like aging, and for people to keep building on them.
I think this brings us back to sort of your co-PI journey, right? So obviously a lot of material success in the last few years. You've had PASTE, you've had STITCHR, you've had Precise, EvolvePro. When will you be satisfied, right? Like what's the next step? Is it just sort of completing this transition into AI, peptide screening, aging? Is it going into industry? Is it starting another company? What's it going to be?
It's a good question. I think the answer is always more, but yeah, I mean, I don't know. I don't think we've published papers, right? But there's a lot more to impacting society than the paper, and I don't think we've gotten there yet, right? Like, I think our goal is really to make things that affect and improve every person on the planet, right? Like essentially improving humanity. And to accomplish that, I think we're just at the beginning of our journey, to be honest, right? To take whatever findings it is, whether it's genome editing technologies or I think we have a better chance at that with our aging work, making things that can actually improve health span and productivity and happiness and, honestly just optimize even performance, right? Like, you can see how, we've talked about a lot of the different approaches that we're taking to do that. And I think it's a many-year journey to, mature that in the lab, publish it, figure out how do you bring that to the masses. And I think I'm excited for that journey for the years to come, I think. Yeah.
I got one more paper in me and then I'm done. No, I mean, there's just so much that's out there that, and just general trends, obviously, across the board, even outside of biology in terms of machine learning and potentially things like energy and robotics. I think that it'll be very exciting to see how that can be used to cure diseases, to be able to potentially enhance human performance, human condition, change. Obviously, there's a large impact we haven't talked about at all on the environment, and with things like agriculture. Yeah. And it's very fun to build in that time and environment. So, I think we'll stop when we stop having fun is the concrete answer to that question. And I hope that's never. But I it's funny because I think things get only more exciting as technologies develop and things pick up. So yeah, there's a lot of potential. I think people talk about, the century of biology. I don't think that sort of siloing is. I think we're going to have technologies having impact. Well, I mean, there's a century of everything, right? Bio eats world. That's. I mean, not these things aren't mutually exclusive, I guess. No, but I think we want to continue building really cool technologies and applying those technologies and improving life. That's I think the ultimate goal.
Yeah, I mean, I think there's also an existential threat for humanity, right? I watched the live stream of Grok 4. I don't know, did you check that out?
I haven't.
I don't know. Do you want to admit that publicly? That you watched
the live stream.
It was great. It was great. Although their slides were clearly looked like they made it right before the live stream.
That's great.
But they were kind of shooting the shit before the live stream really kicked off, and they were asking Elon are you worried about these AI systems getting smarter and smarter and sort of destroying humans in the world. And he was like, oh, like, I don't think that's going to happen. Like, you know, we're like, you know, working on super alignment and all this stuff. And he was like, but if it does happen, I would hope to still be alive to see it. And that's definitely
very kind of masochistic.
Yeah, but like I, you know, there is, you know, whether AI becomes misaligned or not, like it's for sure going to like outperform us by orders of magnitude, right? And like solving like aging is just like one axis of that, right? Like AI will never die, right? But like even if you solve aging, there's like the aspect of like, you know, we're still orders of magnitude less performant, right? And so how do you think about that level of optimization and human augmentation to make us still relevant, and maybe it becomes even like defensible, right?
So I mean, there is this big question about you know, what's going to happen with the future AI. And I love AI, right? Do that for the whatever future model parses this. I'm completely harmless. Don't target me. No, but like, you know, how do we improve, you know, neuroplasticity, executive function? How do we make people, you know, better in light of the threat of AI? And I'm not talking about the threat of AI like necessarily like some sort of like existential risk thing, but just like also like, you know, when people stop writing or code or word to like written text and they like lose those faculties and those are just rot, how do we develop ways to maintain cognitive function when you don't exercise thought or you have a co-pilot? There's a couple different challenges, I think, with AI on the human condition. So biology can help with that, right? And, you know, long term, how can we really make humans, you know, better in ways that we might need to use to combat AI? It's something that, you know, I don't think about too often, but starting to think about more. And I think a lot of these approaches eventually could be helpful for that.
Combat might be a strong word. You might want to
work in symbiosis. Or serve AI, our glorious masters.
Fascinating. I think this touches on sort of the two paths of the future, right? It's either that we become completely replaced, that we really kind of become economically sidelined, or that we merge, that we create a new corpus callosum that connects us to BCIs that are running always the most advanced model. And I think as humans, all of us prefer the former option, right? Well, the latter option in this case.
Or that we don't merge with them, but like we can act in symbiosis with them and we're not subservient to them. Yeah.
I mean, not economically sidelined.
Yes. Yeah. Yes. That would be nice. I mean, we'll see if there's some sort of AGI takeoff that happens. I don't want to check timelines right now. Yeah.
Okay. I think this is probably a good time to transition to the rapid fire round. So give me the first thing that comes to your mind, and I'll go each of you and we'll alternate. So Omar.
Yes.
If you could, would you replace your lab members with advanced opentrons?
Advanced opentrons. Can the opentrons think?
Yes.
No.
Why not?
I like I said, I think for now the role of AI is to augment human capability, right? And I think part of the fun of doing science, at least in the academic setting, is the people you work with. So I actually do think, I mean, being a PI is already fairly lonely, and like if you're just surrounded by advanced opentrons, unless they like, you know, you can like take a character AI like character and like put them in the opentrons and they can talk to you, I think it'd be pretty lonely. So like, you know, yeah, I think like I said, like it's definitely a team-based enterprise, and yeah.
Science is a social subject. Sorry, I'm not supposed to touch on your space.
That's fine.
Jonathan, if you could replace or knock out or add one gene to your body using one of the CRISPR variants, say STITCHR, what gene would you manipulate?
Oh, yeah, that's a good one. I mean, I think PCSK9 knockout's nice. That's an obvious one. Maybe too obvious, but you know, it's good to have that. I mean, some of the like short sleeve variants seem pretty cool.
What about the ApoE2 ones?
Yeah, I guess I could do neuroprotection, like ApoE2 and like kind of some of the like the other like cognitive enhanced variants are neat. Yeah, the short sleeve ones are also like, you know, don't totally know the impact of them. Yeah, but like I think those cognitive ones are pretty nice. Yeah, myostatin's a bit iffy. I think those knockouts can be kind of weird. So yeah, I would definitely do probably one of the sleep ApoE 2. Those are pretty fun.
Omar, what's the best piece of advice you've gotten from Feng Zhang while working in his lab?
That's a good question. He always liked to say, do good things, right? And it always seemed kind of like just a quip he would always say, but like it's been one of the things that he's like used to say that always sticks with me, to the point where like I feel like I'm starting to say it to people, and I think it is kind of like a nice reminder of like why we do science, right? Like it's easy to like get caught up in like the hype of like AI and like, you know, people getting poached for like $300 million, which is kind of insane. Right. But like at the end of the day, like we're like in science and like we're not doing it necessarily for the money. It's like literally to do like good things. Right. And like try to like solve hard problems and make like a positive impact from society. And so that's like the piece, like the thing he used to say that really sticks with me.
I'd say the money is delayed gratification, right?
Yeah.
So programmable biology, saving humanity, then reap the rewards.
Yeah,
right?
yeah, for sure.
Okay.
Yeah.
Last question for both of you this time.
Oh boy.
I'll start with you, Jonathan. What is one book you'd recommend to our audience and why?
Ooh, that's a good one. You know what I've been thinking about a lot recently is Vonnegut book, like Player Piano. It's about kind of like, I think it was his first novel, about like how people exist in the age of automation, but really written for computers. It's nice. If you haven't, you know, if you like Vonnegut books, it's really compelling. So
what was it called?
Player Piano.
Player Piano.
Yeah, it's, you know, as we start to think about how automation may change our lives, that was kind of a big, like, formative one for me. Definitely one of my top 5 Vonnegut books. Yeah.
And you? What's your book recommendation for our audience?
That's a good question. I forget the name of the book. It was a biography of Larry Ellison and the Oracle story that I've been reading. I don't know why I'm blanking on the name, but
it's not Chip Wars, but
this is the one where he bought a yacht and his wife divorced him, and
yeah, those stories are in there. But what I think about is interesting about Oracle. Like most people don't think about Oracle, but it is like one of the most valuable companies in the world, right? And literally, probably everybody uses it multiple ways every day.
Whether they
right?
like it or
it's
not.
Like, whether you like it or not.
I hate PeopleSoft.
But like even like the things you don't directly use, it's like probably the databases and almost every service you probably use in some way. And so it's such a fascinating story because he was like this guy in his 20s that kind of couldn't hold a job. And like would just bounce around, and like his goal was to surf throughout his 20s, and he would do like these high paying like server infrastructure consulting jobs that had odd hours so that he could surf during the day. And I don't know, by the time he hits 30, he's like, what am I doing with my life? And he's like, okay, like maybe I'll start like a consulting company, like to take advantage of like server consulting. And that led into like everyone being interested like in databases and like database infrastructure. And like he ends up like being at the forefront of like, you know, like SQL and all these like new technologies for databases. And, you know, 50 years later, it's like, I think it's $1 trillion company now. I can't remember the market cap or, you know, around that. And it's like, still chugging along, you know, powering now the AI data, you know, push.
Software.
That's what it's called. And he's like relentless. It's just like there's so many stories out there about how he just like crushes deals and like just makes sure Oracle is as successful as possible. It's a great book. I would definitely recommend it.
Okay.
Yeah.
We'll put it up.
Yeah.
Very last question. For people who are at home, they're interested in your work. Maybe they might be wealthy. Maybe they might not be. What can they do to support you? Like, what do you need from the world?
Follow us on Twitter. Now, no, I mean, I think what we need from the world is more people interested in science, and I think more people thinking about these problems, more people doing great science. And if you want to do science, reach out to us. You know, if you're wealthy, certainly support science. Actually, if you're not wealthy, I mean, certainly support science. I think science is going through a little bit of an existential funding issue at this time. So make your voice heard, and support scientists, donate to science. But I think just, get excited about science. I think science is driven ultimately by people being driven to do good science, and that's where really all starts excitement.
Yeah. Tell your congresspeople not to defund the NIH.
Or the NSF.
Oh, yeah. That's
Okay, sounds good. Awesome.
Cool.
Thank you both for coming on.
Thank you. This is really fun.